So let's move on with the last talk of this session. For the last talk, we have David Emms from the University of Oxford, and he's going to talk about OrthoFinder, building a fully phylogenetic orthology analysis. Welcome, David.
Okay, thank you very much. Can you all hear me?
Yes.
Yep.
Yes.
And let me just share the screen. And screen one, share. And can you see the slides now as well?
Yes, we can.
Perfect. Okay. Well, thank you everyone for coming to the talk today, and thank you especially to Rosa and to Jesus for the invitation. It's really nice to talk to so many people working in comparative genomics, and also slightly intimidating, but let's never mind that. Orthology is central to comparative genomics. Orthology basically tells us what we need to compare with what. And so we have a couple of important concepts. The first of those is orthologs. So orthologs are genes descended from a single gene in the last common ancestor of those two species. And then we also have orthogroups, and orthogroups are just a generalization of orthologs to multiple species. So an orthogroup are the group of genes descended from a single gene in the last common ancestor of more than two species. And basically, these are the units we want to use when we're doing comparisons across multiple species, just like orthologs are what we want to use for pairs of species. And so when we came to develop OrthoFinder, I think what motivated us was that the methods available at the time for orthogroup inference really weren't as accurate as they could be. So I've got here an example of one complete orthogroup in the plants, and I've shown with different colors the multiple different sets that OrthoMCL splits this orthogroup into. So you see it split into these 8 paraphyletic sets without giving an indication of the relationship between these genes in different sets. So we have this problem of orthogroups being split up into multiple groups. And then we have the opposite problem of multiple orthogroups being combined into one big blob, effectively. And that occurs especially with genes which have particularly long gene sequences. They all get put into the same orthogroup, and that's incorrect. So we wanted to improve that. And so in our first version of OrthoFinder, we developed this method for better identifying the genes that should be in the same orthogroup and correcting this gene length bias. And we're able to eliminate this bias and produce a method which was a lot more accurate for orthogroup inference and more accurate than the next best method that you could run, which was OrthoMCL. But the nice thing about orthogroups is that they provide the gateway to any kind of other analysis you want to do. So, you know, really what we wanted to do was say, can we take these orthogroups, infer gene trees for them, and use those to infer orthologs? It sounds quite simple, but you know, in practice, there's a number of things you have to take care of and you need to do. So this is the pipeline we developed to actually do that. So we take the orthogroups that we've inferred, we infer unrooted gene trees for those orthogroups. Now we need to root those gene trees, because if you put the root in the wrong place in your gene tree, you'll get your orthologs wrong. And then finally, you need to work out what the orthologs are within the gene tree. And that turns out to be quite a complicated problem if you want to get the accuracy high. So I'll spend a bit of time talking about that. And then in order to actually find the root of these gene trees, you need a rooted species tree. Now, if you've got that, then you can provide that as input to OrthoFinder. But if you don't, then we developed a couple of methods just to firstly infer the gene tree. So a method called stack, which basically said, okay, there's a lot more data in this multi-copy gene families than there is in these single copy gene families for inferring the species tree. So can we use that instead to infer the species tree? We're able to infer them quite accurately with that data. And then our second method was just for finding the root of the species tree based on gene duplication events we observe in the unrooted gene trees. And basically combining those two together got us to this point where we could use the rooted species tree to root our gene trees. And so the next problem we want to address in OrthoFinder is how do we get the orthologs from those gene trees? And the problem is, if you try and read a gene literally, as Celine explained in her talk, these gene trees never perfectly match the species tree. That's why we combine data from hundreds or thousands of genes to try and get the right species tree. But when you've just got the sequence data for one gene, it's very hard to get the gene tree right. So if you try and read a gene tree literally, you're going to massively overpredict gene duplication and loss events and massively underpredict orthologs. And we can have gene trees which are really messy, they look like this, it could be bigger. So we need a good algorithm to sort of interpret these trees correctly. So there are existing methods for doing this. There's a duplication and loss method, which as explained, just over-infers duplications and is too inaccurate. A nice method that exists is what I've called the species overlap method. This is a method that things like PhylogeneDB and Ensemble Compare, these online databases, use. And it's got quite good accuracy. But my concern was that you can make bad predictions just because of a single misplaced gene. And we see an example of that here. I should explain, sorry, the species overlap method says that if the species coming from one side of a node overlap with the species coming from another side of a node, we say, okay, they're gene duplication events. But you can see here, just getting this branch slightly wrong makes it look like the elephant gene is not an ortholog of this wolf gene. So we wanted to avoid that problem. And as Celine talked about a bit in her talk we've just had before, there's this method called DLCPAR, which is one of these more sophisticated methods for looking at this problem, which has this idea of duplication loss and deep coalescence. And we can think of this coalescence in a sense as a rearrangement of this tree that we want to account for when we infer orthologs. And that proved to be very accurate, but just too slow for these large scale, even medium size analyses we want to do with OrthoFinder. So we developed a new hybrid algorithm, which basically took the best ideas from this species overlap and DLCPAR method, and basically used the species overlap to make a crucial simplification in the DLC model, which made it easier to solve. So instead of this large random search through exponentially large tree space, we could just make a single traversal of the gene tree and perform the tree reconciliation like that, which basically gave us a method which was as accurate as DLCPAR, but about 1,000 times faster. And so now we had all the components in place to actually perform phylogenetic inference of orthologs. So we submitted these ortholog predictions to the Quest for Orthologs benchmarking server. And so OrthoFinder are these silver, gold, and black bars here. And on all 4 of the tests, all versions of OrthoFinder were more accurate than all the other existing methods for ortholog inference, and particularly than, say, OrthoMCL, these purple and green bars here. But not only that, it was important to us that we made this method so that it was as fast as possible as well, or as fast as reasonable anyway, because a fast method means not only can you finish your analysis quicker, but it means you can do larger analyses with more data than you could imagine with other methods. And so we're able to make it competitive with the fastest of these BLAST-based methods, these methods that use BLAST scores to infer orthologs, even though we do a full gene tree analysis of the data. And just to give you an idea of the kind of data you get from such an analysis, with OrthoFinder, you get a rooted species tree, and then you get all your genes separated into their separate orthogroups. So you get like 20,000 orthogroups, and each of those orthogroups has a rooted and reconciled gene tree. For every pair of species, you get a table telling you what the orthologs are between each species, between those genes, and it references them back to the gene tree, so you can see the evidence for every single ortholog inference it makes. And you also get all your gene duplication events. So every gene duplication event that occurred since your species started diverging, and they get mapped to where they occurred in the gene tree and where they get occurred in the species tree. And that's really nice, because that means, say you're interested in the ancestor of the vertebrates, you can say, well, tell me all the gene duplication events that happened on this in the common ancestor of the vertebrates, and you can look up in the table and see all these genes that duplicated there. So that was our method that we developed for inferring orthologs from gene trees. And so that was, we published that last year, and I'd just like to talk now about a few updates. I've published or put online a new version of OrthoFinder with some updates a couple of days ago. And this is where we try and say, okay, we can infer orthologs with gene trees. Can we push that back? Can we try and infer orthogroups with gene trees as well? And Yang talked about this yesterday, exactly this problem. Basically, if you've got a large gene tree, you can identify the orthogroups in that by splitting it up. And she described a method for doing this, of inferring extremely large trees and then trying to find the orthogroups within those trees. So it's really a two-part thing, getting the larger trees and identifying the orthogroups. Now, I've been working on a method more recently to try and do this, but with a couple of differences. So I'm trying to avoid making the trees so large that it becomes impractical to do this kind of analysis, because, yeah, you can't have a trees with hundreds of thousands of genes, and you won't be able to do that. And then the second part of this problem, well, I should say the first part I've not yet got a solution for, but the second part I can talk about now, because that's in our latest release. So basically, we recognize that this high accuracy ortholog inference method we had would also do exactly what we needed for orthogroup inference. And so we can use it to split up our orthogroups, split any up that shouldn't be in the same orthogroup, remove genes which are incorrectly placed, and calculate orthogroups. But it also means we can actually calculate orthogroups at every single hierarchical level within our species tree. So we can take this tree and we can identify, well, we've got one vertebrate orthogroup here, but it contains two orthogroups at the tetrapod level. And it also contains these three orthogroups at the mammalian level. So that's in the latest release now. That gives you these hierarchical orthogroups at every single level in your species tree. And by doing this, by using the trees just for this part of the analysis, we're able to substantially increase our accuracy over previous versions of OrthoFinder. So by using the trees to split up these orthogroups, we get an additional 10% increase in accuracy. But also the fact that we get hierarchical orthogroups means that you can now add outgroup species or multiple outgroup species to your OrthoFinder analysis in order to get these larger trees and in order to get yet higher accuracy orthogroup inference. And you can see here that by doing that, we're able to, on the OrthoBench benchmarks, increase the accuracy by 13% over the original OrthoFinder method. Well, I hope I haven't spoken too quickly, because I seem to have got through my slides quite quickly there, but I shall finish here and leave time for questions. I just want to thank Steve Kelly, who's the lab I work in, who's supported me all the way through all this work, and everyone else in the Kelly lab. And thank you all very much for listening. I'll just also point you towards, we've got a webpage of tutorials for OrthoFinder, which show you some of these details and how to examine and analyze the results in more detail. But I'll stop there and take any questions that you have. Thank you.
David, thank you very much. That has been a wonderful talk. Actually, you received so many questions in Slack before today. So I will read the ones that has been written today. I will start by Philip Schiffer asking that, would you mind commenting on Diamond versus NCBI BLAST?
So I think I always universally use Diamond these days because the speed difference is so great and the accuracy difference does not seem to be that big at all. So we have some benchmark results here. The silver one is using BLAST and the black one is using Diamond. So you can see in all cases, the silver one is I would say I think it was 1 to 3% more accurate for final ortholog inference, but the actual calculations are, I I can't remember now, maybe 10, 20 times faster. So BLAST if you want to spend that extra time for that extra accuracy, but Diamond is my default choice.
Okay. So a second question for Felix Thalen is, how does OrthoFinder handle situations where the outgroup is present more than once or where the outgroup is missing when routing the tree? Also,
yeah.
Okay, yes.
Do you want to ask the second question? Because I might have a slide
yes.
for...
How does OrthoFinder deal with the presence of multiple isoforms?
Okay, so in terms of the routing the tree, oh, have I got it here? Yes. So we've got, can you see that slide now? So we have this algorithm, which basically, there are two possible ways you can route a tree. If it's a single outgroup, then you need to route it on the outgroup. Or if it's a gene duplication event, you want to route it on the branch which separates the gene duplicate, those two duplicates. And basically you can work out an analogous score for both of those situations for every single branch in the gene tree using the species tree. And you can work out which is the best possibility of all those cases. So this is the algorithm we use for that. For the multiple isoforms, so in general, I try to put a single isoform per gene into OrthoFinder. I think that's the best way of dealing with the situation because we assume all isoforms have the same evolutionary history. But because of the tree-based method, if you do put multiple isoforms on, they should just end up as terminal duplications, so just pairs of genes at the end of the branches in the gene tree. And so we should effectively identify the orthologs between those genes and any other genes. It just makes it go slower because you've got a lot more sequences to compare.
Okay, we have many more questions. I will put one more from BG Shibulal. It is possible to identify horizontally transferred genes using OrthoFinder?
Yes, there is some. So there is a folder of phylogenetically misplaced genes. I call them that because they are genes which look like horizontal gene transfers, but we don't know, are they transfers or are they contamination or what? So it identifies those cases, mostly in the most extreme cases, and you can look at in there for potential horizontal gene transfers. But its main purpose is actually to just improve the ortholog inference accuracy by not being confused by these genes. So we haven't tested it particularly on its accuracy for horizontal gene transfer exactly.
Okay, we have another question from Alexandros Vasilikopoulous that asks, how do you incorporate uncertainty in inference of ortholog that is due to gene tree estimation error like low signal? Yeah,
so I mean, that's the major problem that we are trying to deal with in this, looking at the gene trees. And it's this model where we basically use this idea from the DLC PAR where you can effectively make rearrangements of the gene tree if it reduces the parsimony score for the total number of duplications, losses, and coalescences that you see. So if one small rearrangement gets rid of a whole load of unnecessary duplications and losses, you say, this looks more plausible, and we take that as a best interpretation of the gene tree.
Okay, we have another different attendee, Arun Prasanna Nagarajan. Does OrthoFinder completely remove deep paralogs while inferring orthogroups?
So paralogs from duplications before the origin of the divergence of the species in question, I assume is the question. So the original OrthoFinder algorithm could sometimes put those together. And so one of the main purposes of this phylogenetic inference of orthogroups is to actually split them up and to separate them. And yeah, that was what I was showing at the end, which it only came out a couple of days ago, but it does that and increases the accuracy quite a bit as a result.
Okay, thank you very much. I'm sorry for the pending people. I will just allow one more question from Leonardo de Oliveira Martins.
After the talk, I'll also go onto Slack and try and answer as many as I can.
Thank you very much, David. You're really kind. How do the benchmark performance is calculated? Since it seems to be based on the species tree, but we know that the gene tree should not always follow it.
Yeah, I have problems with those because so OrthoFinder specifically tries to identify cases where it looks like an orthogroup, an ortholog, but it doesn't match the species tree. In those cases, OrthoFinder will actually be penalized for predicting that ortholog, even if it is correct. I think the benchmarks are very good. They're the best ones we have at the moment, but we really need some expert curated orthologs from gene trees to give us a better idea of the accuracy of all of these methods.
Thank you very much, David, and thanks to the rest of the speaker. So we are going to stop for a bit. Now we will come back in 7 minutes to 2:15 in here and in Barcelona's time. Yeah, see you in a bit.